Good day. In this series of videos, we will discuss the pathways involved in immune signaling and its applications in disease and management. In the first video, we will review the features of innate and adaptive immunity and briefly talk about cytokines and their receptors. Then, in the second video, we will talk about the proteins, cells, and receptors involved in immune signaling and discuss how these signaling pathways activate and regulate the effector response. And finally, on the last video, we'll talk about an example of its application in disease and management by discussing the pharmacology of drugs used in HIV and the use of BCG vaccine in type 1 diabetes mellitus. The dynamic interaction of cells of the immune system with other cells, antigens, and secreted factors determines the nature of an immune response. The response of the individual cell is governed by the sequence of intracellular signaling events triggered following the association of cell surface molecules during cell to cell contact or the detection of soluble molecules of host or pathogen origin. Now let's dive into our discussion points and review the features of innate and adaptive immunity. Innate or non-specific immunity is our first defense against invaders. The epithelial and mucosal linings of our respiratory and GI tracts, as well as our skin, are major components of the innate immune system. These act as physical and chemical barriers against pathogens. Cells known as macrophages also play a role in innate immunity. Macrophages recognize bacterial or viral components such as lipopolysaccharide or double-stranded RNA via special receptors known as toll-like receptors. The adaptive immune response is mediated by immune cells known as your lymphocytes, and these are your B and T cells. The B cells secrete antibodies highly specific for protein molecules that bind to a specific pathogen. These antibodies bind specific parts of pathogens known as antigens, either presented extracellularly on infected cells or free floating in the body. Antibody binding attracts mechanisms that will then attack and destroy the infected cell or pathogen. Some of these B cells become memory cells, which help the body remember the disease and prevent reinfection. T cells can either be helper T cells or cytotoxic T cells. Helper T cells help activate B cells, attract macrophages, and secrete cytokines. Meanwhile, your cytotoxic T cells create pores in infected cells through which they introduce chemicals that trigger apoptosis, thus actively killing the cells. Now, your regulatory T cells, formerly known as your suppressor T cells, are a subpopulation of T cells that modulate the immune system to maintain tolerance to self antigens and prevent autoimmune disease. Now, comparing these systems side by side, innate immunity is first of all an ancient system present in most organisms. The receptors of the innate immune systems were developed during the evolutionary process. The presence of similar structures in plants, invertebrates, and invertebrates, including mice and humans, confirmed this idea. In contrast, the receptors of your acquired system developed only in vertebrates as a result of the rearrangement of genes. There are also differences in the pathogen structures recognized by both systems. The receptors of the innate immune system recognize conserved structures present in large group of pathogens, for example, your lipopolysaccharides in all gram-negative bacteria, while your receptors for the acquired system recognize single epitopes expressed on a single pathogen. The effectory functions of the innate system are non-specific. They are connected with the cytokine-dependent antiviral and antibacterial states of the cell, with pathogen phagocytosis and with the production of cytokines, especially your IFNs, TNFs, ILS, and chemokines, and it involves the killing of infected or transformed cells that is dependent on natural killer cells, macrophages, DCs, and the complement system but independent of your MH. The cells engaged in both systems are also different. And now let's briefly talk about your cytokines. Cytokines are secreted by immune cells in response to cellular signaling and bind to specific membrane receptors, which then signal the cell via second messengers, often tyrosine kinases, to alter cellular activity or through gene expression. Now, yung interleukins comprise the largest class of cytokines and are manufactured by one leukocyte to act on other leukocytes as signaling ligands. Cytokines are often produced in cascades. We have different cytokine receptors which are named after their ligands, such as your hematopoietin family receptors, which would include your colony stimulating factors. We also have your interferon family receptors, your tumor necrosis factors, and your chemokine family receptors. Over the years, some cytokines have been developed into protein therapeutics using recombinant DNA technology, and this would include your bone morphogenetic protein used to treat bone-related conditions, erythropoietin or EPO used to treat anemia, and granulocyte colony stimulating factor used to treat neutropenia and in cancer patients, interferon alpha and beta used to treat multiple sclerosis, and several more others. And that ends this video. Stay tuned for the next to know about pathogen recognition and immune signaling.